Morgan. How are you?
Good. Hi, David. How are you?
Good. Great to have you here.
You too.
Thanks for coming along. You're actually my first guest where I have a name for the podcast. It's very creatively called the Agingdoc Podcast, but at least it has a name now, little
Yep.
logo. So welcome. It's great to have you. So I thought I would give you a little outline for my thoughts in terms of this interview. Right now we have one is introductions, which we're in the midst of right now. Early beginnings, a little bit on how you kind of got started in the lab. It was really hard. You had so many great publications to choose from. So we'll feel that out,
Yep.
And after that, there's some specific things we'll go into in terms of methylation clocks, their uses.
Sounds great.
All right, let's do it. I wonder if you could share with me the process by which you identified this aspect of aging biology in the first place, how you found Steve Horvath that early on. I mean, these were really early days.
Yeah, so I would say I'm not someone who from a very early age knew I wanted to be a scientist. I kind of random walked and stumbled upon it. And I think a lot of it was I found a topic that I thought was extremely interesting. So studying aging. So I did my PhD actually in aging science, so in gerontology at USC, where they actually have a whole school on gerontology, both social science and biological sciences. And then after that, I ended up doing, actually, it's quite a short postdoc with Steve. So I only was with Steve for about 2 years.
That was an impressive list of publications out of a 2-year stint.
I tried to crank them out, and actually, ironically, totally off topic from science, but I ended up I was pregnant right when I joined Steve's lab, which is also bad timing, a computational scientist versus the bench scientist. So I was actually ironically going to go back east for a postdoc, and then due to family things, I ended up needing to stay in California, and Steve was across town at UCLA, and I had worked on biological age measures before, mostly using clinical measures. So it was, a good fit. It was, as you said, early days for epigenetic clocks, and kind of fun to be there, not at the very beginning, but pretty close to the beginning. When I was at UCLA, I applied for what's called a K99R00, which is this kind of transition award for postdocs, and ended up getting that and moving to Yale, where I was there for, I believe, 5 years before getting recruited to Altos. And again, I feel like things that were completely unplanned. I pictured myself in academia for the entirety of my career, and things changed when, this opportunity arrived, and I felt like I couldn't say no to it. It just,
and for the audience, I did not implant that as a segue to the transition. I love your use of the word random walk, because we both understand the statistical implications of that. True to your roots. I was thinking that the 2-year period with Steve was rather formative, and it probably worked out for the best for your career. I mean, it's for you to reflect, not me. There's something to having just enough exposure to a paradigm, but not to be so long in there that you necessarily get entrenched to.
Yep.
I mean, I think it's possible to overcome that potentially, but the fact that you kind of left the nest early on.
Yeah, I think it also depends on where we define what a clock was. So actually, I just learned recently the Wolf-Reich had a methylation signature in the 90s that was associated with age. So, there's some debate. I think some people think the first clock was 2011 by Bocklund et al., which Steve was an author on that, but not a senior author.
Yeah.
And then the Hannum clock actually came out about a year before the Horvath clock did. So
he was the first to popularize it, I think, the way
for sure. I think he made the field what it is in terms of really bringing it into mainstream.
It's like the calculus, like Leibniz gets no credit whatsoever. It's all noon, and so sometimes it's personality, sometimes it's timing, sometimes it's finding the right application for things to have legs, or just for the field in general to be ready. But I see you as the pioneer, not just all the progress you've made on your own right since that time, but you really came in so close to that specific inception. What I think about is the ground zero for at least aging biology community and really taking off. It's been exciting watching your career unfold. And by the way, I could also relate to the story you're talking about. Saying you were pregnant at the time, and I was kind of talking like, oh yeah, I remember being at Brown where I did my residency, and that's when our son was born. So it was just, I can't compare it with motherhood, but it was just insane still.
Yeah, exactly.
Especially, this is really a great segue, I thought, to the next stage in your career. Like, when people make major moves, even just like changing their like a PhD advisor or something like that, less default to have a sort of academic career already rooted, already in a track, and it would have been very easy to build that up. But and then there are all these sort of ideologic countercurrents too. Like, I don't know if this played a role for you, Morgan, but I remember in college, one of my professors, and I even got like from the department chair, Ike Bush, letter of recommendation. I hadn't met him before, but apparently when I met with him, he had been talking to the other faculty members, and they were all saying I should go into academics, and what a waste if I wanted to be a doctor, like I expressed I was interested in, because doctors, they're just technicians. So that was funny, and actually it was also funny because I went full circle. I ended up getting the PhD, and then I kind of iterated back and forth where I ended up doing private practice. But if you could tell me about that process for you in terms of that shift and what's happened since.
Yeah. So, as I said, it was completely accidental. I was extremely happy at Yale. Things were going very well. I was well funded. I had a fairly large lab. I was about to go up. I was preparing to go up for tenure. So it was kind of like all these things happening at once. And then I had been to this meeting with a bunch of the people who had kind of started Altos, and then got a call that they were actually going to start this new, I don't know if I call it necessarily company. It's almost like something more, but this life science institute to some degree. And would I join? And, when I heard about them, I'd known already about the mission and kind of the leadership, who I completely respected and, loved the opportunity to be around them. It was a hard decision because, the academia, once you have tenure and things like that, there's a lot of stability, and you can be there forever for the rest of your career, but you never know with these brand new things what they're going to become, or, what their longevity is. But I felt like I would kick myself down the road if I didn't say yes to this. And of course I had, a family, and there was all that negotiation, but yeah, I don't regret it for a second that I said yes. And it's been a very exciting ride.
Well, I think everything that's unfolded since that time has spoke for itself in terms of the decision you made. And my only wish of you is, like, in your heart and over the long term, for it to continue to be like that. But certainly with a proposition like that, I'm not comparing it to my little puny, like my little moonshot for today was basically I went from anonymous since about 2020. I picked today before, like just a few hours before we interviewed. It's like, why not? Rip off that Band-Aid, know what it's like. Will I have any regrets? I don't know. And certainly being, I enjoyed being private, but I thought, I'm always going to be wondering if I could make more of an impact. And it could be, little things like, oh, it would be cool to, which to me is big, like, oh, like to use my longevity medicine knowledge to help just a few people, versus they say, hey, but then people find out anyway. And now it means I could start a podcast and do other stuff and maybe help promote the field of value of translational geroscience and its underpinnings in aging biology. Yeah. So that's kind of the anecdote I have to share in terms of making, like, I've kind of iterated back and forth. Most of the places I applied for out of residency were actually academic programs. There were 2/3 of the places I applied. So I was surprised when I found like a private practice that had an academic feel. Maybe it was a little bit like that
Exactly.
for Altos for you. Yeah.
Definitely made it easier for Altos. Yeah, they really did try to take parts of academia. And as you can see, most of the people who came are from academia. So what are the parts we love about academia that we want to keep? And then maybe what are the parts that you can borrow from industry that'll make, science accelerate and more exciting, and maybe a little less incremental, which I think has been a little bit of a plague in academic science.
Yes, it's so I have followers and people listen to this from all walks of life, and I think my main takeaway is people sort of know what can figure out for themselves what their own moonshots are, and it's about having an environment where you thrive most. I mean, I think people I don't think anybody should be like a physician, for example, like a doctor, unless they really, really enjoy taking care of people. It's fun. They're a people person. They like helping, and I think it's the same thing. You kind of naturally find your niche, and sometimes little experiences can move you in one direction or another. Like you were at a real formative time, right? You were deciding, the tenure track. I remember one of the tracks for me when I decided to apply to med school for sure. And this was before I discovered, I could do my PhD in health services research. This poor guy, he's like a prestigious faculty. I'm going to kind of call him out. David Goldfarb, if you're still out there in University of Rochester, you were an aha moment for me. You were one of the most intelligent, inspirational professors ever. One day, I was walking down the hall and I saw you, and you were kind of like almost swooned over, like leaned against the wall, and then kind of collapsed down to your bum on the ground. And you had a piece of paper in your hand, and you looked astounded and extremely relieved. And I asked you, "What happened? What happened?" And he said, "I got my grant. I can't believe I got my grant." And I thought, if someone so brilliant as David Goldfarb is thinking about and stressing over getting a grant, anyway, so we all have our moments, and I take it you've been enjoying. It really shows.
Yep. Yeah, it has been fun, especially again being there at the ground floor and getting to build it with everyone else.
Yeah, segue to some of the publications. So we'll be doing publication, and we'll go into as little or as much detail as we want, so that because we got plenty of pubs.
Hopefully, I remember all of them.
It'll be a gift. No, it's okay. So it could be it can be a one sentence or whatever. Yep. But I thought a great way to orient the audience, particularly those who don't work with methylation clocks or even just omics-based biomarkers in detail for their own lab. One paper that I thought had a very, very succinct overview was your assessment of epigenetic clocks as biomarkers of aging in basic and population research. In this publication, as discussed in the primary paper by Nelson et al., which was discussed, developing biomarkers of aging would serve three important purposes. And that's my segue for us to discuss those. One, biomarkers can provide both diagnostic and prognostic insight that may inform medical and or personal decisions. This is probably the only thing that biohackers out there are thinking about. And it's a real issue in terms of personalized medicine, like how reliable we can make them, how predictable. Second, they may uncover mechanistic clues that aid basic research, which is absolutely vital if we're going to make progress. And third, they have the potential to inform intervention strategies, which if people were to said, you're only allowed 3 key issues in geroscience, definitely it's the top 3, maybe number 1, along with some other structural things in the translation aging. So for those 3 purposes, can you give us a little overview about your thoughts about where we were, where we are, and where we need to go?
Yeah, so I think we are not quite where we need to be for at least the existing biomarkers to be so kind of have enough construct validity that we actually could use them all the time for these things, especially I think for the first one, I think it was the first one you mentioned, where it's about informing personal or medical decisions. I think they can give us hints maybe to what those decisions should be, but I guess my worry right now in the field is how reliant people have kind of become on the exact numbers they get out of some of these measures. And, the issue is, which one you use will give you a different answer. And, overly kind of prescribing your life to based on one single measure is a little bit concerning. That being said, I think they do give us a general idea of kind of a spectrum of health. So you can, to some degree, understand within a population how relatively healthy people are using these measures. And from that, maybe that'll change in terms of kind of primary or secondary interventions and prevention. So there's some aspect there. And I think they're getting better and better. And I think if you can combine a few and kind of come to the same conclusion across them, then that helps. I do think actually perhaps a less explored area, but I think is quite exciting, is how they can help us understand the basic biology and mechanism, and how they can actually be applied in research beyond kind of epidemiological population research. So research at the bench. And I think that's really where we're to some degree going now. So how do you understand what these measures are even a proxy for, what they're telling us, what feeds into them, and how? And then this naturally goes into kind of the last point, right? Like, how do you intervene? Once you understand how you can manipulate this state, how can you use them to test? But again, with the caveat that these aren't perfect readouts. And, I see way too many papers say, oh, we had an intervention and we saw a decline in the epigenetic, that they always say the epigenetic clock. And I always think, well, which one? The epigenetic clock by, X number of years.
Methylation clock.
Yeah, exactly. And then that means we reverse staging. So I think that is taking them again too far. But I do think it's where you can start getting early kind of proof of concept that maybe an intervention is going in the right direction, but then it needs to be backed up by a lot of other evidence, so more functional readouts or other types of readouts as well.
That's a thoughtful response. Later on, we'll get deeper into the weeds in terms of mechanistically. It's intriguing, the prospect that how reliable these clocks can be from not just the first generation clocks, which was like chronologic age, but actual mortality and other hard outcomes. Mm-hmm. And they can sort of be tweaked towards different outcomes depending on what we're interested in. And that's a whole other can of worms. Like, how do we have the right set of specific and generalized and non-epigenetic and other metrics towards validation in trials? But even if we just focused on the epigenetic clock aspect, it's intriguing on the footsteps if this came more development in epigenetic reprogramming and other modalities to try to implement whether you can get into a younger epigenetic state, set point. There are also more fundamental questions that I don't think have been fully resolved, in that truth be told, our epigenetic change by some metrics is greater than our genomic instability over time across a lifetime. And if we mess with the epigenome, we see accelerated aging, and we know that the epigenome writes the script for DNA expression patterns across our lifetime, including those that correlate with biologic aging process. What we don't know as well is how specifically CpG sites used in this way, to what extent that represents a highly predictable stochastic process, that's one, or a stochastic process that highly tracks to outcomes we care about, versus some of those other more fundamental mechanistic things, which we know to be the case partially, but how much and how do we tease that apart? I wonder philosophically if you've moved in one direction or another, teasing apart the tracking versus the being. In epigenetic clocks, you also can be measuring compensatory adaptive changes. That could be why it tracks more with that. And where are you now compared to where you were 2-3 years ago?
Yeah, I mean, I think I'm of the mindset that actually what's captured in epigenetic clocks is a mix of a few of these things. So, epigenetic clocks don't necessarily just capture one type of kind of methylation change or one flavor of methylation change, and they're really just a composite of lots of different patterns of change that are happening. My hypothesis would be that some of those, and actually I have some idea about which ones are this random stochastic process. And actually, it seems to some degree like the random stochastic ones are actually perhaps less impactful for kind of future health outcomes. It seems like that. It's not conclusive yet. And then I would say the other kind of big signal, it seems like to me in the clocks, are more programmatic changes, but not programmatic as in like an organism is programmed to age and die, but more the cell's programmed response to things that are happening over the lifetime. And many of these responses actually can become maladaptive over time. So as they become kind of entrenched and chronic. And so I feel like a lot of the CPGs in clocks seem to be more along those lines. And those seem to me to be the ones that are actually more informative for things like health and kind of biological manifestations of aging.
We'll leave that there. Excuse my throat. Among my adventures, I may have a minor case of COVID. So if I'm clearing my throat, I apologize. My MA had it, and we all wore masks, but like right after I got sick, and I tested, I'm negative, but
okay,
who knows? We all know about the issues about sensitivity, specificity, receiver operator curve, less to follow all the different variants. This is a light one, probably a quick one, I'm going to guess. But from a systematic review of biological, social, and environmental factors associated with epigenetic clock acceleration, this was pretty comprehensive as of the time, and this was 2021. And among the areas explored was gender, BMI, physical activity, socioeconomic status, alcohol use, smoking, HIV infection, as well as frailty, cancer, cardiovascular disease, diabetes, lung function, COPD, schizophrenia, and depression. I wonder, what have you found most surprising in terms of what does and doesn't correlate with pick your clock of choice? It depends which clock.
Yeah, I mean, for the second generation clocks, the ones that are kind of morbidity, mortality predictors, I would actually say that nothing is super surprising. Everything that comes up are things that we already know are bad for our health. And in that way, it's like the epigenetic clocks are just another indicator. You can throw them in instead of, remaining life expectancy or disease risk and low socioeconomic status, BMI past a certain point, smoking, inactivity. All these things are things that we already knew. And actually, this always gets to the point when people ask me, "Oh, how do you keep a young biological age?" And these are basically the things I tell them: don't smoke, exercise, eat well. And people don't like that answer. They want like some secret they haven't heard before. But right now, that's mostly what we have. It's lifestyle, behavioral things that we kind of already knew. Some of these things are not within our control. So your socioeconomic status there's a lot of kind of barriers to increasing that for some people. But I mean, some of the behavioral stuff, we do have more control over it than we like to think.
That's a great answer. And I'm a little biased, coaching and consulting in longevity medicine, lifestyle medicine. And that's all about getting good sleep, lowering stress, exercise, diet. It's music to my ears, your response. To be clear, I have tremendous excitement, enthusiasm for, and even the need for us to develop these clocks is one of the most pressing areas in aging biology. Having said that, if we look at level of validation, it's the risk factors for these chronic diseases and lifestyle risk factors that are most validated at this point. So they're kind of the gold standard, but they're not very precise. And what does a good diet really mean? And a lot of it's observational data when our trials are very short term. And that's why we need these. So although I'm saying lifestyle medicine is most important in terms of personal decision making over the short term, I'd like this to see an era where, not by itself, but in concert with functional markers, traditional laboratory other risk factors that these methylation clocks and other omics-based clocks, both specific and generalized, can really give us information much sooner in a way that we can quantify and tune. So tip of the hat, salute to you for your response.
You do bring up a really important point, right? Like right now, a lot of the lifestyle advice is very standardized as well, right? And where we would ideally want to go as these clocks get better is that people could investigate more personalized kind of regimen. So, for me, what type of exercise and how much do I need to actually get, and you can actually make kind of cost benefit decisions. If I add an hour of exercise a day, how much more does that give me? And is it worth it to me? And I mean, the clocks are unfortunately not quite there yet, but I think that would be, in the ideal world where we could finally get to.
Adequately granular and reliable data at a personalized level so we can surf the efficient frontier of optimized health based on our bioindividuality as well as our personal preferences and likes. A lot of listeners here, I'm sure, are familiar with cost-effective analysis.
Yeah,
and that's you can really just have to define the variables. And I try to get into the controversial, interesting, but without sensationalizing either. But
Yeah.
I'll just want to give a tip of the hat to those who have unconventional moonshots in terms of where we would like to be going. There will be controversy always about where we are now, but I like the idea of optimizing with what we have now while being grounded in relative importance and confidence in the different variables. So that's what all this research is all about.
It's a nice example of maybe where I think we can go, and it's good that people are willing to kind of put themselves out there.
Among the areas I found interesting is around tracking, because I asked you about tracking. One of them is menopause accelerates biologic aging. So this is interesting. You first authored this, and you can incorporate data since that time also. But whenever you have a study that comes out, you're going to have individuals who see it through a different lens, both from a researcher lens and from a clinical lens, and at different levels of proficiency in terms of interpreting its implications and drawing inferences. So without feeding the witness, what do you think about possible causality here? And what bearing do you think this has, doesn't have, et cetera, for hormone replacement therapy and other forms of hormonal therapy in other settings?
Yeah, so this is actually, I think this might have been the first paper I did in Steve's lab. So I think, when I joined Steve's lab, he basically gave us the Women's Health Initiative data set and said, look at whatever you are interested in. And so I picked this because I had actually worked with Tuck Finch at USC, who kind of ingrained in me that to some degree, fertility, reproduction are intrinsically tied into aging. So I was kind of interested in this question. We're also looking at kind of early fecundity, but that didn't quite shake out in the analysis. I actually felt like the title was a little strong in terms of asserting causality here. Again, it was an observational data, and I think if it were solely up to me, I probably wouldn't have said anything about causality in this paper. There was a little bit of evidence that maybe that was the direction, because we had data on oophorectomy, so kind of surgical menopause that seemed to also accelerate. But at the same time, there are reasons people are getting oophorectomies. They're not, you're not randomizing people to that group. So I feel like we should have been a little cautious. I would say that this would not be a basis for people to go out and say they need to get hormone replacement therapy as they're going through menopause because they want to slow their biological aging. And actually, I think we looked at HRT in this paper, and it did not seem to benefit in terms of blood epigenetic clocks. And I think, there are things around like hormone replacement therapy. This, again, not my exact expertise, but, the timing of it seems to matter. The duration of it seems to matter. So I wouldn't say that people should draw too much from this, except what I would draw is that we need to pay maybe more attention to women's health and how these kind of things like reproductive aging actually could be entangled to some degree with aging and kind of other biological aging factors. But that would be my conclusion from the paper.
It's a super complex issue, and I think
Yeah.
it speaks to the importance of work like this and different designs to try to answer exactly those questions, those hypotheses. So many layers of controversy. Like, for example, in aging biology, you learn early on about the insulin IGF-1 pathway and how too much of that too early is bad. And then the more you look at the data, the more complex it gets. It's like, okay, this, not just from a like crude level in terms of body composition, which may or may not always be great surrogates, but introduced later in life, is that just like caloric restriction is more powerful, even rapamycin to a degree in rapalogs, but not as much as caloric restriction. Caloric restriction, not as much as more direct genetic interventions along insulin IGF-1 pathway that you have a disproportionate effect that's earlier on. So yeah, I think it's a big open question, and I find it always interesting hearing from researchers reflecting on their own work, and it speaks to the importance of more work. I wanted to transition us actually, Morgan, to like another paper.
Yep.
So we were discussing, the original paper, 2 papers back, we're discussing like surprises or interesting things. Something else interesting. Now, you work in statistical and other modeling, so you appreciate all too much, more than most, the importance of multivariate models are based on a ton of variables, and one might track one way, others track another way. So I like this title. Is 60 the new 50? Okay. Examining changes in biological age over the past 2 decades. Yeah. And truth be told, the first comment is that lifespan has been extending over that period of time, as one expect, not tremendously, and we're not talking about maximum lifespan insofar as we're measured, but more mean. For some, this might be a little counterintuitive because of although we have less heart disease, for example, now, and I don't think better treatments for cancer plays as much into it, but like for heart, but other sorts of preventative care. But now we have this huge obesity epidemic that's just been growing and growing and growing. And how much is this or is this not telling us about the impact of, obviously the way to answer about BMI is just to measure BMI. And if BMI is too crude because there are other things contributing to your BMI, like how much muscle you have, things like that, then your visceral fat and so forth. But I wonder if you could comment about the finding about that despite the obesity epidemic, what's going on? What are your own hypotheses about their relative importance, and how you might think about next steps, disentangling that a bit?
Yeah, so this is a paper actually from my PhD, and here we did biological age was not an epigenetic clock. It was actually one of these clinical markers I'd worked on during my PhD. And actually with the reviewer, we got in a kind of back and forth on we shouldn't be calling it biological age because it's, not, it's just some proxy. But yeah, I think what we found is that there was a decrease in this measure or estimate of biological age during, I think here was about a decade and a half we looked at. This is really testing my memory because it was about a decade.
Dig up that data, Morgan. Can you dig up that data and now apply the best second plus generation clocks to that?
No, so this for this we actually had to use NHANES data, which is nationally representative sample. They do actually now have DNA methylation to some degree. It's not publicly released, but I don't know if they have it at time points that would allow you to look. So NHANES, they go and they randomly sample the population. So you don't get the same people tracked over time. It's just every, wave is a different random sample of the population. We assume it represents the same kind of random sample.
Yeah. Let's get into some exciting controversy. Want to get into some exciting controversy?
Yeah.
Okay. One of the most controversial publications is low protein intake is associated with a major reduction. It's like, yeah, so IGF-1 cancer overall mortality in the 65 and younger, but not older population. I keep this intellectually critical, but I tend to let those invited discuss their narrative. So if you could discuss what you found, the methods, the critique of it, and your current thought about it.
Yeah, so this is actually another one of my papers from my PhD. Here I worked with Walter Longo, who many in the aging field know, and it's been very involved in kind of understanding nutrition and earlier work on caloric restriction. Yeah, so we actually use the same data set again. So this is NHANES. So we're assuming this is representative of at least the US population. I won't make claims about the rest of the world. Caveats are it's an observational data set. People are self-reporting what they eat. So, it's not a randomized clinical trial. So, you can't make complete conclusions. But basically what we found is that we looked at kind of the percent of calories coming from protein versus carbohydrates versus fat, and how that was associated with risk of future things like all-cause mortality, cancer, diabetes, heart disease. We controlled for as many kind of covariates or potential confounding variables as we can. So I think, I don't remember the entire list, but almost anything we could think of that could be potentially confounding here. And basically what we found is first that there seemed to be some interaction. So we actually looked at with age, whether there's an interaction between protein and there seemed to be, and it seemed to be kind of this, about 65 was like this point where it kind of crossed over. And so the results suggested that high protein intake, so the highest protein group had higher risk of things like, I think it was diabetes, all-cause mortality, and cancer. But that this switched in late life, and actually the higher protein group seemed to have less risk. We did what I would call a poor man's version of a mediation analysis. We didn't have, time points. So we just had to kind of say what proportion of this is accounted for by a variable. And it seemed to be that IGF-1, at least we had circulating IGF-1 levels, seemed to matter. So if you had high IGF-1 and you were young, high protein was problematic. But if you were older and your IGF-1 will naturally drop as you age, that the protein actually got it back up. So it's almost about kind of keeping IGF-1 in this kind of sweet spot and how protein kind of modulated that. There was some data on yeast and mice to back up some of the claims we had. So it wasn't purely observational, but yeah, this is my first experience with a study that got tons of attention. It was actually, as a PhD student, it was very overwhelming, the amount of attention this got. I was getting emails and people were saying, yeah, all sorts of things all over. Most people who actually didn't read the paper had lots of comments. So like, oh, it's probably, they didn't account for this. And I was like, you can literally go to the paper and see that. It was a nice, getting thrown in the deep end of what kind of working in these hot topics. So I have to say too, I am thankful for Walter here, because usually like the PI, they do all the interviews, they want to, handle all the press and not, but I feel like he actually gave me an opportunity, which was weird, being just a PhD student to, talk to the press and discuss things. So that was an exciting kind of first moment of that. Yeah.
You learn a lot. You really learn a lot. Both the details, but also human nature, psychology,
Yeah, exactly.
talking points. It's fascinating, really.